Imagine your immune system. Like, it's supposed to be your high-tech defense force, right? Your personal Iron Man suit protecting you.
Mm-hmm.
But then something goes wrong. Instead of fighting invaders, it starts attacking you. In rheumatoid arthritis, or RA, that civil war targets your own joint.
Exactly. And, for decades, the big challenge in treating these chronic autoimmune conditions has been stopping that friendly fire. We have drugs that suppress the whole system, sure, but there's still these huge gaps.
Right, like what specifically?
Well, two main things really stand out. First, how do we actually prevent RA in people we know are at high risk, stop it before it even starts?
Okay.
And second, for people who already have RA, how do we stop those awful, unpredictable flare-ups, the ones that cause so much pain and lasting joint damage?
Yeah, the flares are brutal. So today we're going to dig into some really interesting research that offers a potential solution, kind of like a nanoscale surgical strike. It's aimed at retraining the immune system's command structure.
That's a good way to put it. This work comes from researchers like Wade T. Johnson, Elizabeth L. Wilkinson, Neha Iyer, and their colleagues. The corresponding authors are Nunzio Bottini at Cedars-Sinai's Cow Autoimmunity Institute and Nisarg J. Shah from UC San Diego's Department of Chemical and Nanoengineering.
Okay, quite the team.
Yeah, and they published this in ACS Central Science back in 2025. They developed this new polymer nanoparticle system called Agg-CLNP.
Agg-CLNP, got it.
And the idea is pretty elegant, actually. Instead of just, carpet bombing the whole immune system, they want to create something specific, maybe even off the shelf, that actively teaches the immune system's key players to tolerate the body's own tissues again.
Retraining instead of just suppressing.
Precisely.
Yeah.
Because fundamentally, RA is this chronic inflammation driven by the immune system. And while our current drugs, the DMR's disease-modifying anti-rheumatic drugs, have made a huge difference.
They have.
Getting people into a long-term, flare-free remission, that's still really, really hard.
So if RA is this military operation gone wrong, who are the key players, the generals, as you put it, that need reprogramming?
Okay. We're talking mainly about dendritic cells, or DCs.
DCs.
Think of them as the critical intelligence officers. They're antigen-presenting cells. Their job is normally to find a threat, like a virus, and show it to the T cells, the soldiers telling them what to attack.
Okay.
But in RA, they mistakenly identify parts of your joints as the enemy. So these DCs present joint tissue to the T cells, kicking off and sustaining the inflammation, mostly in the lymph nodes near the affected joints.
So if you could just tell the DCs to stand down.
That's the idea. If you can convince the DCs to stop issuing the attack orders, the whole damaging process could potentially grind to a halt.
And that highlights a big limitation with some current treatments, doesn't it? They often focus on the T cells, the soldiers, but the DCs are still there, basically shouting, attack!
Exactly. Take a drug like abatacept. It's a common biologic DMARD, technically CTLA-4 IgG.
Right.
It works by blocking a kind of molecular handshake that T cells need to get fully activated. And it does help studies show it can delay the onset of RA in high-risk folks, sometimes for 18, maybe 24 months.
That sounds pretty good.
It is, but here's the catch. It's often just a delay. The underlying programming of the DCs, the attack order, hasn't changed. So eventually the benefit can wear off, and many patients still progress to full RA.
Okay, so that covers the prevention challenge. What about the flares? People often get corticosteroids for those, right?
We do. Corticosteroids like dexamethasone, they're like the emergency fire extinguishers. They work fast to reduce symptoms during a flare, but they're a blunt instrument, a sledgehammer, really. They don't usually prevent the next flare, and using them repeatedly comes with a whole host of serious long-term side effects, like osteoporosis, diabetes, thinning skin. It's not ideal for chronic management.
Right, so they put out the fire temporarily, but the house is still wired to catch fire again, and the extinguisher itself causes some damage over time.
That's a perfect analogy.
Yeah.
They don't fix the underlying immune misprogramming.
So this Agg-CLNP nanoparticle, the goal isn't just to block the handshake or put out the fire, but to actually change the orders given by the dendritic cells. Tell us about the nanoparticle itself. How's it built?
Okay, so it's pretty clever engineering. The base is a biodegradable polymer called PLGA-PEG.
PLGA-PEG. Heard of that.
Yeah, it's widely used in drug delivery because it breaks down safely in the body over time. No accumulation. And the particle is tiny. We're talking less than 100 nanometers.
Wow, really small.
Super small.
Yeah.
And that size is crucial. It's optimized so that when injected, say, near a joint, It naturally flows into the lymphatic system and travels straight to the lymph nodes close to that joint.
Where the dendritic cells hang out.
Exactly, right to the command center.
Okay, so it gets to the right place. What's it carrying? What are the instructions?
Two key components. First, on the outside, it's decorated with something called an immunodominant aggrecan peptide. Let's just call it Ag. Okay. Aggrecan is a protein found in cartilage, in joint tissue. And this specific Ag peptide is known to be recognized by the immune system in people at risk for RA. So it acts like a address label or a homing beacon for the DCs involved in the autoimmune response.
So it uses a piece of the enemy signal to get the DC's attention.
Precisely. And then encapsulated inside the nanoparticle is the second key ingredient, calcitriol.
Calcitriol, that's the active form of vitamin D3, isn't it?
That's the one. And calcitriol is known to be a potent immunoregulator, especially for dendritic cells. It can push them towards a more tolerant, less inflammatory state.
So it's like a Trojan horse, using the Ag signal to get inside the DC's sphere of influence and then delivering the calming calcitriol payload.
That's a great way to think about it.
Yeah.
They tested this in the lab on human DCs from both healthy people and RA patients.
And what did they find?
Really consistent results. The Agg-CLNP particles significantly dialed down the inflammatory signals on the DCs, specifically molecules like CD40, CD80, and CD86.
Remind us what those do?
Sure. Think of CD40, 80, and 86 as the go signals. They're co-stimulatory molecules that DCs display on their surface when they want to activate T cells. By reducing these, the nanoparticle essentially tells the DC, hold fire, don't activate those T cells.
Okay, so it dampens the attack signal.
Yes, but it does more than that. It also actively upregulated a molecule called CTLA4 on the DCs themselves. CTLA4 is generally known as an inhibitory molecule on T cells, but here, seeing it upregulated on DCs suggests the nanoparticle is actively pushing the DC towards an immunoregulatory, a tolerance-promoting phenotype.
So it's not just stopping the bad signals, it's promoting the good ones too.
Exactly. It's actively reprogramming them towards peace.
And one really crucial finding you mentioned earlier was about localization, right? Avoiding systemic side effects.
Yes, this is huge. When they injected Agg-CLNP into the muscles of their RA mouse model, they checked the levels of calcitriol floating around in the blood plasma systemically. Nothing. No change. The nanoparticle treatment delivered the calcitriol effectively to the local lymph nodes near the joints, modulating the immune cells there, but it didn't cause a spike in calcitriol throughout the rest of the body.
That's critical because high systemic doses of calcitriol can be toxic.
Absolutely. This targeted delivery gets the therapeutic effect exactly where it's needed in those lymph nodes where the autoimmune response is being orchestrated without the risks of systemic exposure. That's a major advantage for potentially developing this into a long-term therapy.
Okay, so the particle works on its own. Now, what happens when you combine this internal reprogramming with an external blocker, like abatacept? They tested this in the SKG mouse model, right? Which mimics high-risk pre-RA patients.
They did, and the combination was, well, synergistic is the word they used. It was much more powerful than either agent alone.
How so?
The combination of Agg-CLNP plus abatacept significantly delayed the onset of arthritis and dramatically reduced the severity compared to just giving abatacept by itself.
So abatacept puts the brakes on T-cell activation, while Agg-CLNP is retraining the DCs. Together, they're much better.
Much better. Think about the clinical scores they measured, like a measure of arthritis severity. At day 14 in the study, the group getting just abatacept had a score around 2.6. The combination group, their score was down around 0.58.
Wow, that's a huge difference. Much less inflammation and disease.
A very significant reduction. And importantly, it wasn't just masking symptoms. They look at the joints under the microscope histology. The combination group showed much better protection against actual joint damage. Significantly less synovitis, that's the inflammation in the joint lining, less bone erosion, less loss of proteoglycans, the stuff that makes cartilage spongy.
So the synergy actually protects the joint structure long-term.
It appears so in this model, yes. It suggests the combination is truly modifying the disease process itself.
Okay, that's the prevention angle. What about controlling flares in established RA? This is the second big challenge. They used a dexamethasone withdrawal model for this.
Yes, exactly. This model mimics what happens when a patient uses corticosteroids like dexamethasone to quiet a flare, feels better, stops the steroid, and then the inflammation often comes roaring back, or relapse.
Right.
So in this model, they first treated the mice with dexamethasone to induce that low disease state, that temporary quiet period. Then they gave the Agg-CLNP nanoparticles.
And what happened when the dexamethasone effect wore off?
The Agg-CLNP treatment significantly reduced the severity of the subsequent flare. The mice that got Agg-CLNP after dexamethasone maintained much lower clinical scores compared to mice that just got dexamethasone alone. It really helped prevent that rebound inflammation.
That's pretty impressive. And you mentioned something about durability and steroid tolerance.
Yeah, this was a really compelling finding. When they gave repeated injections of dexamethasone alone over time, the drug started to lose its effectiveness. The mice seemed to develop steroid tolerance, which is something we see in human patients too.
The drug just stops working as well.
Right. But, and this is key, the group that received Agg-CLNP after the initial dexamethasone cycles, that treatment kept working. It retained its effectiveness through repeated flare cycles, measured out to day 42 in their study. The scores stayed low.
So the nanoparticle treatment provided a durable control that the steroid alone couldn't.
Exactly. That suggests it could be a way to maintain remission and prevent those recurring flares long term, potentially avoiding the need for repeated and less effective steroid courses.
What's going on under the hood? What's the mechanism for that sustained flare control?
It comes back to those dendritic cells in the local lymph nodes. The Agg-CLNP treatment seemed to preserve that tolerant immunomodulatory DC phenotype even after the dexamethasone was gone.
Keeping the generals calm.
Keeping the generals calm.
Yeah.
And importantly, this had downstream effects. They saw suppression of specific types of T cells called Th17 cells, both in the nearby lymph nodes and actually in the paws, the site of inflammation.
Th17 cells, why are they significant in RA?
Th17 cells are notorious in RA. They are potent drivers of inflammation and are strongly linked to the bone and cartilage destruction you see in the joints.
So, by keeping the DCs calm, Agg-CLNP indirectly suppresses these really destructive T cells locally.
That seems to be the mechanism, yes.
Okay.
And they did a crucial control experiment. They made nanoparticles that were identical, except they removed the Agg peptide, the targeting signal. Those nanoparticles, lacking the Agg targeting, had no protective effect against the flares.
So that proves the targeting, getting the calcitriol specifically to the relevant DCs via the Agg peptide, is absolutely essential for the therapeutic effect.
Absolutely confirms it. The targeted delivery is non-negotiable for this to work.
Okay, this all leads beautifully into the molecular deep dive. They used RNA sequencing to look at the genes inside these reprogrammed dendritic cells. What did that genetic fingerprint reveal?
Right, the RNA-seq gave him a snapshot of exactly how the Agg-CLNP was changing the DC's behavior at the genetic level, and it painted a very clear picture of a shift towards tolerance.
Okay, tell us about the genes that were turned up, the peacekeepers, maybe.
Yeah, peacekeepers is a good term. They saw upregulation of several key immunoregulatory genes. SHEL4 was up, confirming that regulatory push we talked about earlier, even on the DC itself.
Okay.
Also, CCL22, which is a chemokine, a signaling molecule that's known to attract regulatory T cells, or TREGs.
TREGs are the immune system's own peacekeepers, right?
Exactly. So the treated DCs are basically putting out signals saying, hey, TREGs, come over here. We need to calm things down.
Yeah.
They also saw upregulation of CD200, another marker associated with tolerogenic DCs that dampen immune responses.
And you mentioned one other critical one, TSC22D3.
Yes, TSC22D3. This gene codes for a protein called GILZ. And GILZ is a really important anti-inflammatory transcription factor. Its main job is to suppress a major inflammation pathway called NF-κB.
NF-kappa B. That's like the master switch for inflammation in many cells, isn't it?
It really is.
Yeah.
So by delivering calcitriol, which is known to work through GILZ, the nanoparticle is effectively hitting that master switch and turning down the inflammation at a fundamental genetic level.
Wow. Okay, so it's boosting the peacekeepers and directly interfering with the inflammation command pathway. What about genes that were turned down? Were any pro-inflammatory genes suppressed?
Yes, they found downregulation of a gene called CARD14.
CARD14?
Yeah, CARD14 acts as an immune amplifier. It's also involved in that same NF-κ signaling pathway, but it ramps up the inflammatory response.
So the nanoparticle is turning down the amplifier while turning up the suppressors.
Exactly. It's actively rebalancing the DC's genetic programming away from inflammation and towards tolerance.
So zooming out then, this work from Nunzio Bottini, Nisarg Shah, and their teams, published in ACS Central Science, it feels like more than just testing a new drug particle. It seems like a proof of concept for a whole different strategy for RA.
I think that's fair to say. The first big takeaway for me is that Agg-CLNP demonstrates a really novel, localized way to potentially treat RA by actively reprogramming those key immune cells, the dendritic cells, towards tolerance.
Creating tolerance, not just broad suppression.
Right, that's the goal.
And the second key takeaway seems to be the power of synergy. When you combine this nanoparticle approach with existing treatments, like abatacept for prevention or even short-term corticosteroids for flares.
The results are dramatically better. It suggests Agg-CLNP could offer significant synergistic benefits, boosting prevention, and maybe providing that durable flare control that's currently so difficult to achieve, especially without long-term toxicity.
It really points towards a future, doesn't it?
It does. This kind of research supports moving away from just broadly hitting the immune system systemically and moving towards more precise, maybe even off-the-shelf, nanoparticle therapies designed to induce specific, localized immune tolerance right where the disease is happening.
Okay, so that leads us to our final thought, a provocative question maybe. The research showed that Agg-CLNP works best for flare control after the inflammation is already dampened, like after using dexamethasone.
Right, it needs that initial quieting to establish the long-term tolerance effectively.
But dexamethasone has those systemic side effects we want to avoid long term. So the question is, what other, perhaps novel and less toxic combination therapy could be used first? What could get patients into that necessary low disease state, setting the stage perfectly for something like Agg-CLNP to come in and provide that safe, durable, long-lasting tolerance?
That's exactly the question researchers will be tackling next. Finding that optimal, less toxic induction therapy to pair with these tolerance-inducing nanoparticles
Yeah.
that could really revolutionize how we manage autoimmune diseases like RA.
A really fascinating glimpse into the future of immunology.